about genotype versus phenotype. Your genotype is your genetic code of your cells. This genetic code is responsible for many of your traits. The physical expression of your genotype is your phenotype. Your genetic information is stored in your chromosomes on sections called genes. A gene is a portion of DNA that codes for a trait. The following picture illustrates this. If you look at this drawing of a gene, the different sections are your genes. In genetics, you encounter three types of genotypes: homozygous dominant, homozygous recessive, and heterozygous recessive. For each trait, you get some information from mom and some information from dad. The different forms of the same traits are called alleles. Take a look at the different hair texture. These different forms are alleles. It is still hair, just different forms. A dominant allele will cover up or mask a recessive allele. You will only get a recessive allele if both alleles are recessive. Your phenotype is the actual expression of the genotype. For example, this dog has a round nose. This cat has black hair. These are physical expressions of the genetic code.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous, like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase, and poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Fed bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells.
Epigenomic. Describing a representation of the gene expression, DNA methylation and histone modification status of a particular genomic region.
Today we're going to be talking about epigenomics. Let's start off by breaking down the key part in the word epigenomics, genome. Your genome is your complete set of genetic information that you inherit from both parents. This genome is included in every cell that you have, and it is made of segments of DNA, called genes, that follow the genetic code. The genetic code also directs the specialization of cells. One way this happens is when certain proteins or chemical groups attach to a gene. Once attached to the gene, they chemically modify the gene's DNA. However, they don't alter the genetic code. These modifications then turn on some genes and leave other genes turned off. This process is what makes specific cells, like stomach or heart cells, in your body. Scientists track the modification of DNA for each unique cell type, which is what the cell's epigenome is. But when a cell's epigenome is disrupted, genes can be incorrectly turned on or off, which can cause diseases like cancer. This poses to be a huge problem. When cells are cancerous, they rapidly divide, and the cells being divided carry the same abnormal signals that direct the genes incorrectly. Now that we've learned about the genome and the epigenome, understanding what epigenomics is will be a little bit easier. Let's look at the word parts again. Epi means above, so epigenomics means above the genome. In other words, this includes your genetic code and the elements that define how that code is used. So, epigenomics is the study of the modifications made to the genetic material of a cell. We've already talked about what happens when a cell's epigenome is disrupted, but what does all this mean for scientists? Well, the significance in the discovery of the epigenome is that the DNA code is not the whole story for why abnormalities happen in the body. It helps explain certain phenomena, such as why identical twins can end up with different diseases, even though they have the same DNA. Because genes can be chemically altered without modifying the genetic code, this explains why one twin could get a disease and the other one doesn't. Even though they have identical genes, one twin's gene could be turned on, while the other twin's gene could be turned off. So, they have the same genome, but different epigenome. Let's go back to cancer. Cancer develops either when p53s, which are tumor suppressor genes, are abnormally turned off, or when oncogenes, which stimulate cell division, are abnormally turned on. Instead of trying to kill cancer cells completely, scientists are now attempting to change the instructions of the cell by trying to turn on the p53 gene, or turn off oncogenes. This new approach is called epigenetic therapy, and is just one example of why the discovery of the epigenome is monumental in the science world. Diseases like Alzheimer's and autism are also thought to be linked to a disruption of the epigenome, and the discovery and development of epigenomics could lead to new scientific discoveries to help cure these diseases, as well as many others.
The study of this genetic punctuation that I'm talking about is called epigenetics, which literally means above genetics. The epigenome doesn't change your DNA, but it decides how much or whether some genes are expressed in different cells in your body. Epigenetics looks at what happens to your genes over the course of your life and whether those changes could be passed down to your children or even your grandchildren. So here's the way epigenetics works. You have billions of cells in your body, and they each contain your DNA, the same exact blueprint of your genetic code. But just because they have the DNA doesn't mean that they know what to do with it. They need outside instruction from these little carbon and hydrogen compounds called methyl groups. The way these methyl groups control the genome is by binding to a gene and saying, "Do not express this gene." The methyl groups bind differently to your genome in a skin cell versus, say, a tongue cell or an eyeball cell. And that is one of the ways that a cell knows that, hey, I'm a skin cell, or hey, I'm an eyeball cell, or hey, I'm a muscle cell. In addition to methyl groups, epigenetics is also controlled by histones, which are proteins that are basically the spools that DNA winds itself around. Histones can change how tightly or loosely the DNA is wound around them. If they're more loosely wound, the genes can express more, and if they're more tightly wound, then they express less. So whereas the methyl groups are more like a switch, the histones are more like a knob. Every cell in your body has a distinct methylation and histone pattern. And that is what gives every cell its marching orders. Think of your genome, the DNA, as the actual hardware of the computer, while the epigenome is more like the software, which tells the hardware what to do. The genome is what's going to be doing all the work, but the epigenome is going to be telling it what to do. So the hardware of your DNA is going to be the same throughout your entire life. But these epigenetic tags do change throughout your life, and they decide what genes get expressed or not. Now, epigenetic information in a cell isn't permanent. It can change throughout your life, and it can be hereditary. And it can change over time, especially when your body is going through a lot of changes, like, say, during puberty.